So I talk about universal biology, and so this may look a new term for most of the people here, but actually, this was proposed about 60 years ago by a Japanese science fiction novelist, Komatsu Sakyo, who is a great novelist. And in his novel, so Universal Biology is a science to explore universal patterns and possible variations of living organisms in this cosmos. And it started to expand at the end of last century. And last century means 20th century. And since then, characterization of life in terms of topological geometry, and this means dynamical systems in the theory, has developed. And now grand theory comparable to relativity is anticipated. So this is the novel of 1968. So we need to realize his prediction. And so actually, about 10 years ago, we set up a Universal Biology Institute in University of Tokyo. And so I started there, and now I'm in the Niels Bohr Institute. And so universal biology, as I said, so life system as a kind of universal class in nature. And to make some kind of, for me, to make some kind of general theory for that. And so that means not necessarily true to the life on this Earth, maybe some life in some other planet or somewhere else, and also maybe protocell constructed in laboratory. So we try to understand some kind of general pattern across all of these. So, but then we need to understand or try to define what is life so that can be applicable not only on this Earth. But usually precise definition in the science will come later after some kind of theory is established. So maybe it's somehow difficult to discuss the definition. But probably everybody agrees that maybe life system, it can maintain itself, and maybe in some condition it can self-reproduce. So I think most agree with this. But only by that, even for crystal growth, may be regarded as life. So we need something else. And in the NASA's definition, I think, they add Darwinian evolution to this. But Darwinian evolution is a kind of very higher concept compared with these other two definitions. And so maybe Darwinian evolution could be a result of this some axiom of life. And so for me, the important thing that are missing here is that it consists of very, very diverse components. So that's not observed in standard physics or chemistry, because they usually consist of a few components. But in the life, even for the simple bacteria, they include maybe 10,000 or more components. So that's very, very diverse, but still maintain itself and can produce itself. So that's a very strange property. So assuming this kind of diversity and this kind of axiom for life, maintain diverse components and can produce itself. So the question for theorists is that can we derive other universal properties of life? So here I discuss some of the propositions derived from this kind of definition. The first one is hierarchy. So in life system, usually molecule, cell, and cell consists of multicell organism. There is some kind of hierarchical system. So this can be understood that to have some kind of continuous reproduction against parasitic invasion, this kind of hierarchical system is a kind of necessity. So that's one. The second one is that function, information, division of label. So in the present life, we know there is central dogma, and that is genotype and phenotype are somewhat separated. So gene gives some kind of information transmission, and phenotype or protein or catalyst So they work as a kind of function. So somehow, these roles are separated into two molecules. So of course, this may be just a coincidence on this Earth, but I try to show that maybe this is a kind of general property to have this kind of reproduction system continuously. And the third proposition is that some kind of this system with diverse components, there appears some kind of few degrees of freedom that control the growth of this system. So even though there are very many degrees of freedom, 10,000 degrees of freedom and 10,000 dimensional system, but there are few modes that control the behavior, and that is the necessity to tame very complex system. And then related to this kind of dimensional reduction leads to some kind of directed evolution of this system. So in this case, so the life system, we show that fluctuation is inevitable. And with this fluctuation, this kind of directed evolution can be possible. So that's evolutionary fluctuation response relationship. And then another thing is that biological system against diverse environmental change, so they need to adapt to many different conditions. So for that, they need to change, not to change. So that's an adaptation. So I discuss these issues. So before that, why I need to assume diversity? So if mathematics, okay, this is axiom, and I do not need to discuss why diversity, but in science, maybe we need to discuss. And first of all, usually environment is diverse and non-equilibrium. And maybe in that case, so in the beginning, it's many complex biopolymers appear just because there are more diversity. So it's a combinatorial diversity. So that's the discussion Freeman-Dyson, so the kind of famous physicist. And so he discusses complexity in the beginning. So usually we tend to think the origin is very simple, and then the life becomes more and more complex. But Freeman-Dyson discussed in a different standpoint. So initially, it's complex enough. So that could be a solution. And also, there are many possibilities to have this kind of diversity. So to cope with diverse environmental changes or to resist against parasitic processes that I'll talk later. And, or another thing is that, so if there are some kind of resources, and if the simple system uses just single resources, then they usually eat up all these resources. And then it's difficult. And actually, in a simple mathematical model, we show that if there is a limited resource, then transition to diversity occurs. So for instance, we consider some kind of chemical reaction network, so that is called hypercycle by Manfred Eigen. So mutually catalyze and can grow. Xi is catalyzed by Xj, and so this is Xj is catalyzed by Xi and grow using some resources Si. So if Si is sufficient and flow is large, then actually, if we put many different Xi, but finally, the kind of very good efficient cycle, efficient components, a set of efficient components remain. So when the resource flow is sufficient, most efficient simple one remains. But when the resource is not sufficient, there is a transition. And then this simulation, many components exist and make kind of complex structure. So that's a kind of general result in this simulation. And actually, we can understand mathematically. So this is a little bit mathematics, but anyway, if you consider some kind of concentration of Xi as rho I, and if the resource is sufficient, then the growth of rho I is proportional to rho I. So that's exponential growth. And in that case, if the flow, so if some component AI was some flow Si0, so this is the way of efficiency, and Si0 is the external concentration. So the largest one remains in this case. So this is the result for exponential growth. But if the resource is limited, they take up this resource Si, and finally, and they compete proportional to Xi. And then in this case, if the resource is limited, then this kind of equation. And in this case, growth of each component is not proportional to itself. It's just proportional to the flow. And in this case, even though there are different Si, and some Si is larger, some Si is small, this many species coexist. So there is generally a kind of transition to diversity. And in this simulation, so flow of resources and diverse species, remaining species, then there is a transition, and this increases. So in that sense, as long as resource is finite, maybe we can expect some kind of diversity. So now we assume diversity. So then the next question is hierarchy. And this is somewhat general property also. So replication of molecule is catalyzed by some others and mutually catalyzed and to grow. But in this kind of system, generally, emergence of cheetah molecules, cheetah or parasite molecules can be replicated by others, so catalyzed by others, but cannot help others. And so, but if that exists, they use limited resources, but they do not help the growth. And so finally, the system collapses. So that's a big question of this reproduction system. And actually, historically, there are discussions that this can be avoided by assuming compartmentalization. So these molecules are put into a kind of protocell, and then protocell dominated by parasites cannot grow. So in that case, a multi-level evolution appears. And for example, in this study, so we consider some kind of chemicals, R, and then make kind of complex, and then catalyzed by this, and using resources, they can grow. And then we put number of given number of molecules here. And assuming that if the number is larger than some threshold, it divides into two. So there is one parameter, n. So beyond which it divides. And another parameter is that with this revision, the molecular replication, there may be molecular change. So it's a kind of mutation. So by that, catalytic activity may increase or decrease. So in this kind of parasite, that means this activity just decreases. I do not help others, because when I'm helping others, I cannot be replicated. So for molecular sake, it's better to decrease this kind of catalytic activity. So then what happens if we have n and mutation rate? And then we show that, so this is the number of molecules in the cell, and this is the mutation rate of this. And beyond that, molecule level fitness wins. So everybody becomes a kind of parasite and lose catalytic activity, so that the system collapses. But if n is not large, then cell level selection wins, so that it can survive. So in that sense, to be resistant against parasites, we need some kind of hierarchical system, molecule level replication to cell level reproduction. So in that sense, hierarchical system is a kind of necessity. But still, in that simulation, if n is large, parasite molecules accumulate. So then we cannot make a kind of complex cell with many components. So how are cells of more molecules possible? And for that, we assume two types of molecules are existing. And then, initially, these two types of molecules works as kind of information template and also some kind of catalytic function. So they work both. So for instance, in the RNA world, ribozyme can do both, depending on its structure. And then we did this simulation in this previous setup. Then we found there is some kind of symmetry breaking. One type of molecule works as catalyst, and another type of molecule, the other type works as kind of information transmitter. So this is a result for simulation. So we consider P and Q, two types of molecules, and initially they work both of catalyzing the other replication and information translation. So there are both of these several reactions, and they have some kind of parameters here. But after this simulation, when n is very large, then there is some kind of symmetry breaking. One of the molecules work as catalyst. So in this case, P works as a catalyst, so catalyze every other reaction. And Q works as a kind of information transmitter. And by that, so we can avoid this. So Q is a sort of parasite in that sense. But still, Q works as a kind of information transmitter. And P works as a kind of, function. So in that sense, maybe central dogma, so protein works as a catalyst and DNA works as a kind of information. Maybe a general property, even if we go to some encounters or somewhere else. Maybe the different molecules are used, but maybe there is symmetry breaking. So that's this theoretical thing. And we can do this, and mathematically this can be shown as some kind of symmetry breaking in physics. And interestingly, this kind of function information symmetry breaking is somewhat universal in multi-level evolution. And so I talked about this cell and molecule. But when cells accumulate, and then there is a multicellular organism like us. But in this multicellular organism like us, there are separation of germ cell and somatic cell, germ cell, egg, or sperm, and somatic cell, or other muscle cells or some others. So they work as a function, and they just information transmitter. So this separation is general here also. And also in some social insects, society and individual, so queen and worker. So this queen does not work anything, but just information transmitter. So in that sense, this mathematical framework can be applied generally to this kind of hierarchical system. And furthermore, in this case, these work as a function, so they need more. It's better to have more. But these just kind of information transmitter. So it can be just small. And actually, it may be better to be smaller, because if there is only one molecule, one number here, then its change can influence drastically to the next generation. If some mutation to good thing occurs, then it can be good. But if there are too many, for example, if there are too many DNAs here, then maybe they go to in the different reaction, and then on the average, there is no directed evolution. So in that sense, kind of minority, control appears here. So that actually we discussed previously this. So that's kind of general property of reproduction system. And another, so now I discuss a little bit more on evolution. And so system with diverse components. So there are diverse species, but each number of that molecule accordingly cannot be so many. So it's much, much less than Avogadro number. So maybe some molecules, some protein species in a cell is just 10 or something, or sometimes less or than that. So generally, if that number is small, there is large fluctuation. So actually, even for the same cell with same gene, so the protein number, protein concentration fluctuates a lot, and this is log. And actually, this is close to log normal distribution. And this is because for reproduction, some kind of amplification is necessary. So according to that, so even the noise is amplified. So there is large fluctuation. So the question is that if this kind of fluctuation is related to something biology or not. And in physics, we know fluctuation-response relationship. And actually, this was originally studied, proposed by Einstein. And that is a Brownian motion, and it fluctuates, Brownian motion, particle fluctuates. But this fluctuation, that's a microscopic matter, is related to macroscopic response. So that's what Einstein showed. And there is some other, Kubo and many others formulated this. So basically, response ratio against some kind of external force is proportional to microscopic fluctuation without force. So that's this kind of established theorem. Of course, this is established in thermodynamic equilibrium theory. But can we extend this to the problem of evolution? So evolution selection process. So evolution, and especially selection, we make some kind of mutation and select to a good direction. So it's a kind of generalized force. It's a macroscopic force. But as I said, even before that, there is large fluctuation. For example, fluorescence has such fluctuation. So can we extend this theory to have some kind of fluctuation is proportional to this kind of response or evolution speed? And actually, so we can do something. So if we assume the distribution of some phenotypic variable and some genetic control parameter, and if this is mutated, and then how it's changed, and assuming some kind of simple distribution, Gaussian type distribution or something like that, and we can show that this kind of response and relationship. And now we can apply this theory to evolution. And in this experiment, so that's done by 20 years ago, so they try to increase the fluorescence in a protein in a cell, and by mutating this kind of gene to produce this protein. And then initially, this level of fluorescence, and then make mutants, and this is a good one, and select, and select, 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 select. And so after 10 generations, fluorescence increased. And here, the distribution is that isogenic one. So this is not a mute. So for the same gene, so we make clones, but due to noise, there is fluctuation. So you can see if this variance is larger, the step is larger. So initially, this variance is larger, the step is larger. But then later, this variance is smaller, and evolutionary step is smaller. So that means evolution speed and variance, phenotypic variance, may be somewhat proportional. And actually, in this experiment, actually, this is only red points here. So maybe, so this evolution speed and variance is somewhat proportional. And we did some kind of cell model with many, many components and do the simulation to increase. And this agrees rather well for each mutation rate. So looks nice. But then there is another mystery. So in population genetics, there is famous theory by Fisher that's called fundamental theorem of natural selection. And so that says variance is proportional to evolution speed also. But this origin of variance is different. So in Fisher's theorem, so they are not considering the kind of noise, so but mutation. So by genetic mutation, for this gene, it produces this phenotype. For this gene, it produces phenotype, so maybe fluorescence. For this genetic change, this changes. So if genetic distribution is distributed, accordingly, phenotype or fitness is distributed. And if this variance is larger, then evolution is faster. This is somewhat natural because this change is kind of inherited to the next generation. If the good one appear here and here, then this is selected. But in our case, the same gene organism, but just by noise, it goes here. So the next generation producing from this, it may go to this one. So it's a little bit more counterintuitive. So this is established, and it looks like this seems to be also proportional to evolution speed. So then that means this and this should be proportional. But the origin is quite different. This, what I call VIP, is that isogenic phenotypic variance, is due to just noise. So it's a very fast timescale phenomena within one generation. But this G, VG, is that kind of mutation. It changes slowly through evolution. This is due to evolutionary timescale, and this is one generation fast phenomena. But are these really proportional? And actually, we did that simulation again, and it looks like, okay, VIP and VG proportional. And so another mystery appears. And so, but that means that genetic change and phenotypic change is somehow discussed all together. So we try to make some kind of phenotype, genotype, two-variable distribution theory, and assuming that these are kind of just two variables, forgetting about this time scale is different. And then just consider that this distribution through the evolution can keep single peaked structure. So it's a kind of robustness. So good one has a peak in this phenotypic space and genetic space and keep this structure. And assuming this structure with some mathematics, we can show that this kind of VG and VIP is somewhat proportional. And depending on mutation rate, and there is some kind of mutation rate maximum where the error catastrophe occurs. But anyway, we have seen this. And actually, to check this, we did some simulations and also some other experiments using some kind of a fish or some kind of insects or something and bacteria. They suggest this is fine. Okay, so maybe this is fine, but still, the mystery remains. Originally, I said that biological system is very, very including many, many degrees of freedom, many components. So it's very, very high dimensional. But the distribution theory I used is just X and A or something like that, just few variable. So how is that possible? How kind of huge dimensional reduction can be possible? So that's the next question. And actually, for that, we need to first recall that molecule cell, so it can grow. When cell grows, all molecules are roughly replicated in the same speed. So there is strong constraint. So that means, so if every molecule has to be roughly, be doubled before cell reproduction. So that means if we consider the growth rate of each molecule, this should be equal to all. So even if you have 1,000 molecule species, mu1, mu2, mu3, mu1,000 is equal. So very, very strong constraint. And from that, with some other kind of condition and some other linearization or something, we can show this relationship. This delta xi is that change of each component concentration, actually log concentration change. And this, and in a different environmental condition you apply, is proportional to this, is equal to this. And mu is that the growth rate of cell. So here, you have many, many components, 1,000 components. But this part does not include any J components. So this is independent of all components. So we can check this behavior by using the bacteria experiment. And they did this kind of thing. And what they did is that they put this bacteria into a kind of many stressed condition. So like osmotic pressure and some heat or something like that. And then they measured actually messenger RNA here. So there are 4,000 messenger RNA species and how it changes, this concentration changes. So this delta X is the change. So there are kind of 4,000 points here. the theory says that this is proportional to all components. So it looks like okay. And also the theory says this is proportional to delta mu, and that agrees with this. So this is partly fine. But still, in this theory, we use some kind of steady growth theory and also linearization and something else. So still some more is missing. And actually, we did some simulation to evolve a cell. So actually, the previous theory just uses that kind of steady state. But we know that the present cell is a result of evolution. So we need maybe evolution leads some kind of expansion of this linearization regime or dimensional reduction. So we need how many points? Okay, maybe. Yeah. So actually, even 4,000 components or many thousand components in this cell model, and then we did this simulation, and actually after, yeah. So since there are 1,000 components, so this is change is 1,000-dimensional space. So each component changes by environmental conditions. It's in this 1,000-dimensional space. And what we did is that here, we put this original state and many different environmental conditions, and change here, changes here. And it's originally 1,000 space. But after evolution, this can be roughly fitted along this one-dimensional curve. So this is basically PC1, PC principal component, 3-dimensional, but roughly very low dimensional. But before this evolution, it's just scattered. So after evolution, high, very drastic dimensional reduction occurs. So, and the reason for this, we can expect, is that originally very, very high dimensional. And if there is a good state, but good state is somewhat rare. And then there are many, many noise perturbations. So it can be kicked away by noise, could be. But if it kicked away, this is not good. The good state is rare. So it has to be recovered soon, drastically, to come back. So there is need strong attraction to this good state. So that's one thing. But still, this state has evolved and will evolve. So in the direction of evolution, they need to change. So here, if everything is attracted to this, there is no more change. So there needs to be some direction so that could be changed. So this is some kind of low-dimensional reduction. And that's actually in this simulation. And so from that, we can show this result. And also, yeah, with this environmental change we did. But here, we put some mutation. And mutation says, follows the same line. So along this line, evolution occurs. So even though high dimensional, this reduction to this kind of environmental change and evolutionary change follows the same line. But this also means that the change of this each component by environmental change and mutation follows the same relationship. And also, evolution occurs along this line. So we could check this experimentally, and this is the experiment by Chikara-Furusa's group, and this original growth rate of the bacteria, and they put some kind of stress, ethanol stress, and the growth rate decreases. And then through this evolution, it recovers. And they checked how this kind of phenotypic space changes. And again, using transcriptome analysis of messenger RNA, it's originally 4,000 dimension, but projected to 3 dimension. And then through this evolutionary course, it changes like this. And they repeat this experiment. It is interestingly, genetic change occurs in a different position. So genetic change is somewhat different, but phenotypic change is deterministic. So if you replace the tape, maybe genetic change is different, but phenotypic change is same. So that's kind of interesting result. And this also agrees with the theoretical result of this. And also, maybe this is the last one. So this evolutionary change and environmental change, and also by noise. The change by noise also follows the same line. So that means the change by noise and the change by mutation occurs in the same manifold, same space. So accordingly, we can derive this kind of relationship again. So each component of this variance and each component by noise and by mutation is proportional across all components. So we have totally kind of, fluctuation by noise and fluctuation by mutation and response to evolution, response to environment. All these are somewhat very much correlated or proportional. So that's the basic result. So we showed this kind of proposition first, hierarchical system and functional division of labor. And then there is some kind of consistency, different across different scales. And this kind of, with this kind of structure, maybe slow change along this, and this control this high-dimensional phenotypic change. So, okay, so that is the result. I need to thank this inspiration by this science fiction writer Komatsu Sakio, and also inspiration by Albert Einstein and Freeman Dyson, and biologist Conrad Waddington. And these are kind of more real collaborators. Okay, thank you.